Hey, this is our third video on adaptive immunity. We talked about the different classes of adaptive immunity, and then we talked about the antigens and antibodies that mediate adaptive immunity. So now we'll talk about the cells, and I know we talked about a lot of cells during this lecture or during this unit on lymphatics, but the major lifters or the major cells of the immune system that do the heavy lifting are the lymphocytes, the macrophages, and dendritic cells. These cells are especially concentrated in lymphatic organs, skin, and mucous membrane, and that's to, of course, boost immunity in those regions, and then also protect from bacteria or other invaders from gaining entrance or getting far with that once they have gained entrance. So there are three categories of lymphocytes, which are the natural killer cells, which function in immune surveillance, and then the T cells and B cells. Remember that T cells and B cells are distinguished because of where they grow up. So T cells come from the thymus and B cells come from the bone marrow. Okay, so we can see the life history and the migrations of the B and T cells. So we see from red bone marrow, that is, of course, the sternum, where B cells are leaving and they become part of humoral immunity, and T cells are being educated and grow up in the thymus, and those are generally associated with cellular immunity. So lymphocytes, macrophages, and dendritic cells are huge cells within the immune system. And again, three different classes of the lymphocyte: T cells, B cells, and NK cells, natural killer cells. So with T cells, there are three different stages in the life of a T cell. They are born in the bone marrow, educated in the thymus, and then they're deployed to carry out immune function. So within the thymus, cortical epithelial cells, which are within the cortex of the thymus, release chemicals that stimulate maturing T cells to develop surface antigen receptors. With those receptors, the T cells are now what's called immunocompetent, which means that they are able to recognize antigens that are presented to them by the antigen presenting cells, and they undergo positive selection, which we'll talk about in a second. Medullary, which means in the medulla of the thymus, so the medullary epithelial cells will test the T cells by presenting self-antigens to them. And this is what's called positive selection. So T cells can fail by either reacting to that epithelial cell or reacting to the self-antigen. That would mean that they would attack their own tissues, and that's not really very good. So cells that aren't positively selected for means they fail. So they're eliminated by negative selection. So this could be because of clonal deletion, which means that they're self-reactive T cells and they die, and macrophages will then phagocytize them. Or they can undergo anergy, which is when self-reactive T cells remain alive, but they're unresponsive, and then they kind of just go off and die. So with negative selection, it leaves the body in a state of self-tolerance. So that means that the surviving T cells only will respond to suspicious antigens and will ignore the body's own proteins. I think it's kind of amazing that only 2% of the T cells pass the test. So 98% of them are just discarded. They don't pass the test. And of course, this selection process is pretty strict, and it needs to be so that you don't have autoimmune disorders. So there's a naive lymphocyte pool, which means that immunocompetent T cells, which are able to recognize foreign antigens, but they haven't yet encountered those foreign antigens. So there's what's called naive. So naive T cells are deployed into the body, which means they leave the thymus and they're able to colonize lymphatic tissues and organs within the body. B cells, which are another form of lymphocytes, develop in the bone marrow. So some fetal stem cells will remain in the bone marrow and then will then differentiate into B lymphocytes. B cells that react to self-antigens either undergo anergy, which means they just kind of chill out there, but they're not really doing much, or clonal deletion, which means they are deleted or phagocytized by macrophages, which is the same as T cell selection. So those B cells that are self-tolerant, which means they pass that selection, they are able to synthesize antigen surface receptors, divide rapidly, and produce immunocompetent clones. So that means that they can produce clones that are able to recognize antigens. So these B cells can then leave the bone marrow and colonize the same lymphatic tissues and organs as T cells. So what's the difference between B and T cells? T cells are not able to recognize antigens on their own and require an antigen-presenting cell. Remember, we talked about these antigen-presenting cells. They are able to phagocytize the invader and present the antigen to the T cell. So these antigen-presenting cells include dendritic cells, macrophages, reticular cells, and B cells. The function of these antigen-presenting cells depends on their major histocompatibility, or MHC complex proteins. So these act as cell identification tags that label every single cell of your body that as belonging to you. And these are structurally unique for each individual, so that my major histocompatibility complex wouldn't be the same as yours, except for identical twins that do have the same MHC complex proteins. So antigen processing, which is with antigen-presenting cells, where the antigen-presenting cell encounters the antigen, internalizes it by endocytosis, which is a form of phagocytosis. It will then digest it into different molecular fragments and display relevant fragments, which are the epitopes. Remember, the epitope is where the antibody will eventually bind. So they'll display these epitopes in grooves of the MHC protein and then go to present this to the relevant cells. So wandering T cells will then inspect antigen-presenting cells for displayed antigens. If there is a self-antigen, the T cell will then just ignore it. But if it's a non-self-antigen, the T cell will initiate an immune attack. The antigen-presenting cells then go off and alert the rest of the immune system to the presence of that foreign antigen. So this is key to successful defense, meaning that it can quickly immobilize immune cells against that antigen so that you can get rid of it quickly. So again, with most of the cells in immunity, they require chemical messengers to coordinate their activities. And these are the interleukins, which are a form of cytokines. Remember, we talked about cytokines before. These can be pro-inflammatory or anti-inflammatory. And I think of them as the hormones of the immune system. So here you can see what the antigen-presenting cells do and how they function. So first, they phagocytize the antigen, break down that antigen using their phagosome, and they lyse it. The antigen is then degraded into different fragments. The residue that's not needed is voided by exocytosis, so they get rid of that. And then the processed antigen fragments, the epitopes that are relevant, are displayed on the surface so that they can then bind to T cells. So we'll begin the next lecture with cellular immunity.